All right, so let's reorient here. Down here is the mitochondrial matrix again, the center of the mitochondria. This is the inner membrane, that wavy membrane. Here's the intermembrane space where all of those hydrogen ions were built up. And actually, I should add those back in, right? We had all those hydrogen ions building up from those proton pumps in the electron transport chain. And then I didn't draw the outer membrane because I wanted to have plenty of space to draw on this membrane, but the outer membrane would be out at the top here, right? So that second, the most outermost smoother membrane would be up here. All right, so let's draw those so we learned from the electron transport chain piece that you got all of these hydrogen ions building up here. All right, so you've got this electrochemical gradient. Electro because it's got a positive charge, chemical because, well, hydronium ions, right? That's a chemical. It's actually acid. So this is super acidic actually because acids, pH is a measure of how many hydronium ions are in a solution. There's quite a lot here. So this is quite an acidic environment in this intermembrane space. All right, so this has been, that electron transfer chain has been going on for a while. So you've been building up a whole bunch of protons out here. Now, the reason this is unstable and high in energy is because if these, remember we learned law of thermodynamics, laws of entropy. Entropy is always increasing. Things are, tend to spread out evenly. So if these ions could cross this membrane, they would flow across until they were equal on both sides. Why don't they? Well, they have a positive charge, they're hydrophilic, they cannot cross the hydrophobic phospholipid bilayer. So they're stuck here, but it's almost like you backing up water behind a dam, okay? So when you have a dam, you put this wall in a river and the water's all moving downhill, right? You back the water up behind this wall and the water swells. You're collecting energy behind that dam, right? And you know that you are because you can let that water down its gravity gradient by opening a door or a portal in the dam. You can allow the water to move through in a controlled way. And then you could even harness the energy of the water moving through the dam by, using attaching a water mill to it. And that spinning of the water mill could generate electricity or could grind your grain or whatever it is you need the energy for. And that's the energy in the water moving down its gravity gradient, right? But you had to back the water up behind the dam to really collect that energy for later. You're storing it in the form of water behind the dam. Now, in that analogy, this membrane is the dam, right? This is the barrier. And you've caused instead of water being backed up, you've backed up these hydrogen ions. So they're swelling and bursting to get out. Now, if you could allow them back across in a controlled way, you could harness the energy of their movement. And that's exactly what happens. So let's draw this enzyme called ATP synthase. All right, so ATP synthase is this enzyme that has a few different subunits. So it's got this one piece that's embedded in the inner membrane, and then it's got a rotor. A rotor is something that spins, like on a car. So it's like a rod that spins in a circle. So I'll draw that. It's able to rotate or spin. And then there's this other subunit here that goes around that. So the rotor extends into it, but so that's like inside. Actually, let me try this. I want to make it a dotted line so you can see that it's inside, not extending over the top of, right? So a dotted line of it moving in here. And there's a lump on this inner piece, and that lump is going to open and close subunits on the smaller piece. I'll show you a video because this is hard to describe in an inanimate drawing. It's easier to have a video of, and I do have those. So this little piece is called ATP synthase. ASE at the end of a word, that means it's an enzyme. And enzymes catalyze chemical reactions, right? They facilitate chemical reactions. ATP synthase is an enzyme that synthesizes or makes ATP, right? So that's what we're looking at here. Okay, so and there's this one more piece to this. There's a stator that holds this outer subunit still. So this subunit stays still, or actually doesn't, it rotates. So this subunit is inside, it's embedded in the inner membrane. Here's a rotor that spins, and it extends into this lower subunit, and it's got a lump on it that opens and closes leaves. It's this lower subunit is kind of made of leaves or petals, sort of, and every time this bump passes them, it causes them to open and close, and then close again when the lump passes. And this subunit, this lower subunit, is held still so that it does not spin. So this rotor spins inside this outer subunit that stays still, okay? And this whole enzyme together, it's a very complex enzyme. It's called ATP synthase. This is actually very similar to flagella. So the way that that ATP synthase works, it works very similar to flagella, the way flagella works, which we looked at last week. So this piece is a portal for hydrogen ions to pass through, okay? So hydrogen ions, their chemistry prevents them from crossing the hydrophobic phospholipid bilayer. However, this subunit provides a channel for them so they can flow through. They flow through and out into the matrix, down their concentration gradient, just like all molecules move down their concentration gradient. So this is dissipating the gradient, the electrochemical gradient. As they flow through, this is again like water through a dam. The movement of the hydrogen ions through ATP synthase causes this rotor to spin. So this spins, that causes this lump to spin, opening and closing these subunits. And actually, I want to move this hydrogen ion so I have space over here to draw, because the next piece is spent energy molecule ADP is going to come in. Okay, so you have, when these petals in this lower subunit open, ADP, adenosine diphosphate, the molecule with only 2 phosphate groups, can enter. And they enter with what's called an inorganic phosphate group, which is just a free-floating phosphate group, and we denote it by Pi. So that's just the phosphorus with a bunch of oxygens around it. So that goes in as well. The lump then spins away, the leaf closes on top of these and presses them together. That's like really what happens, and it forces them to react. It forces the phosphate group, another phosphate group, to get added onto ADP. So then it comes out as ATP, adenosine triphosphate, with 3 phosphates. So the molecule only had 2 phosphates when it went in, and it comes out with 3 phosphates, ATP, charged, right? This was the energy depleted version. This is the energy rich version. Now ATP can go and do whatever work it needs to do in the cell, right? It can leave the mitochondria, it can do work in the mitochondria, it doesn't really matter. It's going to diffuse out throughout the cell to do whatever work needs to be done. And these leaves just keep opening and closing as long as hydrogen ions are flowing through this. The spinning will continue to happen, and this reaction will continue forward, and more ATP will be generated. So this piece, so the last part that we covered was the electron transport chain, with all those enzyme complexes that pass the electrons, pump the protons. This piece is often called chemiosmotic phosphorylation. I know that's a big word. Let me write it out here. Chemiosmotic phosphorylation. Other times you'll hear it called oxidative. Well, I know it sucks that there's so many words for the same thing. It really makes things complicated, and I apologize for that. Again, it's not under my control. I really would prefer you to, like I said, I'm usually going to refer to the electron transport chain, plus this piece called chemiosmotic phosphorylation collectively is just the electron transport chain. But you have a textbook that may use these other words, and I should tell you what they are, okay? So the reason it's called chemiosmotic phosphorylation is because this is a chemical gradient. This chemical is osmosing or diffusing across the membrane, and that's driving phosphorylation, or the adding of a phosphate group to ADP to generate ATP. So chemiosmosis, that's the chemical osmosing or diffusing, and phosphorylation is when adding the phosphate group to ADP to create ATP. Right, so that's chemiosmotic phosphorylation. Oxidative phosphorylation, that brings up the oxygen component, right? But I don't like that, when oxygen removed the spent electrons from the electron transport chain. I don't like using that because it's very aerobic centric, and there are anaerobic electron transport chains, which we will cover in the next lecture. There are lots of different kinds of electron transport chains out there. There are ones that use oxygen as the final electron acceptor. There's ones that use nitrate as the final electron acceptor. There's ones that use sulfate or, as the final electron acceptor, or a whole bunch of different kinds of molecules. They all have those enzyme complexes. They all pass electrons. They all pump protons. They all allow protons to flow back through for ATP synthesis. So the process is essentially the same. There's minor differences, but it's essentially the same for all of those different forms of respiration. And so I don't like to use oxidative phosphorylation because it's too aerobic centric when there are in reality many anaerobic electron transport chains that exist in the microbial world. So I would prefer just to use this term, chemiosmotic phosphorylation. Okay. And while we're on the topic of that final electron acceptor, oxygen, so for your cells, it's oxygen, right? So while we're on that topic, I told you that if you didn't have oxygen, ATP synthesis would stop. So when we looked at the last drawing, we looked at how if oxygen wasn't there, the electron transport chain would get backed up. All those electrons, none of them would get cleared. They'd back up and the electron transport chain would grind to a halt. If no electrons are being passed, there's no energy released to drive the formation of this proton gradient, right? The proton pumps stop. If there's nothing to drive the creation of this proton gradient, then these protons will flow back through ATP synthase until they're equal on both sides, and then there will be no net movement anymore. And if there's no gradient, there's no hydrogen flow. So again, think about water behind a dam. If the water on one side is equally as high as the water on the other side, then you remove the dam and there's no net water flow, right? The water doesn't move through the dam. You need the water on one side to be higher, right, than on the other side for water to move forward. And it's the same thing here. You need this chemical gradient to be higher here than here to get this flow to move forward through ATP synthase to drive ATP synthesis. So if you don't have the proton pumps working, if you don't have oxygen, the proton pumps stop. If the proton pumps stop, this gradient disintegrates. And if there's no gradient, then there's no energy to drive ATP synthesis. And I also want to identify these energy transfers. So the movement of protons is going to be kinetic energy. The spinning movement of ATP synthase, that's going to be kinetic energy because it's energy and movement. And so it's kinetic energy that's actually driving the formation of ATP. So energy started as chemical energy in food, chemical energy in NADH, potential energy, a different kind of potential energy in the proton gradient, kinetic energy in the proton flow, kinetic energy in the moving parts of ATP synthase, and then back to chemical energy again, which is a kind of potential energy in the form of ATP. Now it's ATP. Now it can go use that energy for any activity in the cell. So I know that seems like a very roundabout way to make ATP. The reason it might seem clunky, well, part of the reason is it's not you can't just click your fingers and get the energy from glucose into ATP, right? So it requires this complicated process that works not just for glucose, but for many other kinds of high energy molecules. So all fats, other kinds of sugars, they all go through this same process more or less. So that's one of the reasons it has to be fairly complex. But the other reason is just that these things evolve slowly, and components evolve accidentally and in series, and not, it's not like it's planned from beginning to end. At the beginning, there was only glycolysis, right? And then these other pieces came together, usually co-opting features that had other functions initially and reusing them for a new function. And so that whole process just ends up to be a little bit messy and sometimes a little longer than you might imagine it needs to be. But in the end, it's the most efficient energy converting process we have. So the energy lost to heat is something about, I think it's like 66-ish% of the energy in your food is lost as heat. And 30 something percent is is converted to ATP. That may seem inefficient, but it's far more efficient than your car. So your car converts 25% instead of 30 some percent of energy into movement of the car, and 75% is lost as heat. So this is actually extremely energy efficient overall. So I just wanted to show you a couple more formal, like textbook pictures of the electron transport chain instead of my, poor drawings. So here you have enzyme complex 1, 2, 3, 4. Electrons from NADH get passed to enzyme complex 1, ubiquinone, 3, cytochrome C, 4. Electrons from FADH2 go to complex 2 to ubiquinone to 3 to cytochrome C to 4, right? And you can see from this diagram that only enzyme complexes 1, 3, and 4 are proton pumps. It's the movement of the electrons from a less stable to a more stable state, from a high energy to a lower energy state, that drives, that these electrons are donating the energy or giving the energy to these enzyme complexes to allow them to pump protons against their concentration gradient into the intermembrane space. And this sort of orient you here. ATP synthase. So then these protons, there's a lot of them on one side of this membrane, not very many on the other. They flow back through this way, right? And that causes, so there's a F0 subunit, F1 subunit. These are sort of like petals that can open and close. The stator holds this lower subunit still, while the axle or rotor rotates inside of it, causing the petals to open and close.